Good evening, students. So today we'll be learning about beta oxidation of fatty acids. So these are the learning objectives. The learners will be able to understand the steps involved in the degradation of fatty acids in detail. So beta oxidation of fatty acids, it breaks down into acetyl coenzyme A. This occurs in the mitochondria, and the process is strictly aerobic, that is, the presence of oxygen is necessary. And after production of acetyl coenzyme A, it goes to the Krebs cycle. So where does this occur? So it occurs in many tissues like your liver, kidney and heart, and it does not occur in the brain. So there are three steps that are involved in the oxidation. That is, first is beta oxidation, alpha oxidation and omega oxidation. So we'll be studying beta oxidation. So again it has three steps. First is activation of fatty acids in the cytosol. Then these fatty acids are go into the mitochondria using the carnitine shuttle, and then beta oxidation proper occurs in the mitochondrial matrix. So first step is activation of fatty acids. So this process is carried out by the enzyme Fa thiokinase, that is fatty acid thiokinase, also called as acyl coenzyme A synthetase, which is present in the cytosol. So this thiokinase enzyme requires ATP as a source of energy, coenzyme A, which has the group SH attached to it, and Mg2 plus as a cofactor, and this reaction occurs. So the fatty acids plus coenzyme A plus ATP. So the fatty acid and coenzyme A gets attached, AMP is formed, and 2 phosphorus are released. Next is transport of fatty acid from cytosol to mitochondria. So since these are the long chain, there is a carnitine shuttle, which is a special transport mechanism which occurs. So here what happens is the fatty acids get attached to carnitine, it goes into the mitochondria, and then the carnitine is again released, which again carries out new fatty acids from the cytosol to mitochondria. So this is the structure of carnitine, and the enzyme involved is acyltransferase 1, and the acyltransferase 2 brings back the carnitine to the cytosol to carry out further oxidation. So this is the rate limiting step. So the acyl group from acyl coenzyme A is transferred to carnitine to form acyl carnitine, which is catalyzed by carnitine acyltransferase 1 in the outer mitochondrial membrane. Now this acyl carnitine is then goes to the mitochondria by translocase enzyme. The acyl group is transferred back to the coenzyme in the matrix again by transferase 2. So finally the carnitine is released and in exchange of incoming acyl carnitine. Next is beta oxidation proper, which occurs in the mitochondrial matrix. So it has again three steps. First is oxidation of FAD-linked dehydrogenase. Next is hydration by hydrotase, and step 3 is oxidation by NAD-linked dehydrogenase. So this is the palmitoyl coenzyme A. So the beta carbon is one which is present next to the alpha carbon. Alpha carbon is the one in which the carbon next to the functional group is called as the alpha, whereas the next carbon is called as the beta. So oxidation occurs at this beta carbon. So these are the steps involved in beta oxidation proper. So first, your palmitoyl coenzyme A with the help of FADH. So FAD converts to FADH2, and the enzyme involved is acyl CoA dehydrogenase. So here you can see on the beta carbon a double bond is getting formed, and we get trans delta square enoyl coenzyme A. Now your water molecule comes into picture, and H goes here, so it forms L-beta hydroxyl acyl coenzyme A, and the enzyme involved is enoyl coenzyme A hydratase. So as the enzyme is hydratase, that means hydration is taking place. In the next step, NAD is converted to NADH plus H plus, and we get the formation of beta keto acyl coenzyme A. So what change is happening? So here instead of OH and H, it is getting converted to C double bond O. So here is the beta carbon where the oxidation has taken place. The enzyme involved is beta hydroxyl acyl coenzyme A dehydrogenase. Next, the coenzyme A again comes into picture, and we get formation of acyl coenzyme A and acetyl coenzyme A. So again, this will break down into further until all the units are getting converted to acetyl coenzyme. Thank you.